Kingdom fungi. Kingdom fungi is one of a vast kingdom of five kingdom basis classification of living organisms as proposed by Robert Harding Whittaker. Under kingdom fungi, we includes wide varieties of mushrooms. In this kingdom, we are also including one of commonly used fungi called yeast. You know that yeast is used for baking or softening of bread. We are also categorizing one of commonly used fungi called penicillium under kingdom fungi. Penicillium was used for production of antibiotic named penicillin. So kingdom fungi includes wide varieties of mushrooms. We study kingdom fungi in a branch of biology called mycology. So mycology is a branch of biology that deals with the study of fungi. In this video, we are going to discuss about kingdom fungi as part of chapter number 2, biological classification. First, let's discuss in general about fungi. Fungi are achlorophyllous. They are not having chlorophyll. They are non-chlorophyllous living organism. So because of the absence of chlorophyll, they are heterotrophic. They can't synthesize their own food like plants. So they are dependent on others for food. That's why they are heterotrophic in nutrition. They follow heterotrophic mode of nutrition. Fungi are eukaryotic. That means fungi have a well-developed or well-defined, well-organized nucleus, a nucleus which is surrounded by nuclear membrane, and within the nuclear membrane we have nucleoplasm, which is housing genetic material. So genetic material which is enclosed by nuclear membrane is a well-defined nucleus. Along with that, fungi do have cells which are having cell organelles bound by membrane. So membrane bound cell organelles and well organized nucleus makes fungi eukaryotic. Fungi are also multicellular living organism having many cells, made up of many cells, except yeast cell, which is unicellular in nature. Yeast cell is made up of single cell. If we talk about habitat, fungi are distributed everywhere. So they are cosmopolitan in distribution. Cosmopolitan here refers to distributed everywhere. They are found in water, air, soil, even on plants and animals. Usually fungi prefer to grow in warm and humid places. Humid places are having moist condition. Along with that, the temperature is temperate. So fungi prefer to grow in warm and moist places. Because of the distribution of fungi everywhere and their diverse habitat makes them having diverse morphological characteristics. So morphology and habitat of fungi are diverse. Morphology here refers to study of external structures, study of external forms, external appearance. So fungi are usually diverse and they survive in different habitats. Now let's look at cellular characteristics of fungi. We know that fungi are multicellular. They are made up of many cells, and these cells are usually protected by a special cell wall called chitin. Chitin is a polysaccharide. Unique polysaccharide. Unique polysaccharide here refers to these are polymers which are derivative of glucose. Fungal body is usually made up of hyphae. Singular form of hyphae is hypha, And hypha is hair-like structure, thin hair-like structure within which fungal cells are present. So fungal body is made up of or constituted by achlorophyllous hyphae. Hyphae is long, slender. Slender here refers to thin. So hyphae are long, slender, thin thread-like structure. We can also explain hyphae as long, slender, hair-like structures. So hyphae are thin thread-like structure, and many hyphae coming together, joining together, and they form mycelium, which later form a giant fungi, which we can see with our naked eye. So grouping of hyphae or formation of crisscross connection of hyphae will form mycelium, and mycelium, many mycelium will join together to form mushroom. And we can easily find mushrooms growing around us. So mycelium can be explained as network of hyphae. Network of hyphae forms mycelium. Mycelium can be of different types. Some mycelium are septate mycelium. That means within the hyphae of that mycelium, cross walls are present. Cross walls are known as septa, or singular form of septa. Septa is septum. So if cross walls are present within the hyphae of mycelium, then we can say that mycelium as septate mycelium. Opposite of it is aseptate mycelium. Aseptate mycelium are not having cross walls separating the cells of fungi in hypha. So in this diagrammatic representation of hypha, there is no cross walls, there is no septum. So that's why we are calling this kind of, if these kind of, suppose this is a hyphae and similar kind of hyphae form a mycelium like this, then due to absence of cross walls, we are calling that mycelium as aseptate mycelium. Aseptate mycelium can be also explained as cynocytic hyphae. Cynocytic here refers to a hyphae which is having many nuclei. So within the hyphae of aseptate mycelium, we are able to find many nuclei, and these nuclei are not separated by cross walls. So that's why we are calling aseptate mycelium as cynocytic hyphae, for being presence of many nuclei present within the cytoplasm, the common cytoplasm of the hypha. Fungi are good as well as bad. They are our friends as well as foe. Why they are foe? They cause different diseases in plants and animals. Puccinia triticina usually causes wheat rust. Wheat rust is usually observed in wheat crop. So on the stem and on the leaves and parts of the wheat plant, there is deposition of rust-like tiny structures, and those are the Puccinia triticina. So in a way, they cause huge economic loss by infecting wheat. So Puccinia triticina causes wheat rust. In mustard plant, if you observe the leaves of the mustard plants, then you are able to see, sometimes you are able to see white spots. So white spots which are present on the leaves or parts of the mustard plant are usually due to infection by Pseudocercospora capillae. which is a fungi. And on our body also, fungi grows. So because of the growth of fungi named aspergillus in our respiratory tract, these are causing aspergillosis. Similarly, if fungi grows on our skin, uh, then that is due to uh, like this. Suppose if fungi grows in this kind of fashion, then this fungi is mainly that disease like ringworm, uh, fungal nail infection. So fungal nail infection and ringworm. You have seen this kind of appearance commonly during the summer, right? During monsoon. So fungal nail infection and ringworm and aspergillosis are diseases which are caused by fungi. So fungi cause disease, and on the other hand, they are good fungi because they are useful in our life. So penicillium is used commonly for production of penicillin. Penicillin is an antibiotic. So penicillium is a fungi which is used for production of antibiotic named penicillin, which is good for treatment of some diseases, some other microorganisms. Uh, so we also have neurospora. Neurospora is commonly used for different molecular biology studies. So molecular biology study can be biochemical or different genetic engineering work. So neurospora is commonly used for studies in molecular biology. Yeast, which is technically called scientifically called as Saccharomyces cerevisiae, is also having different uses in production of bread, as well as they are also used for production of alcohols, which are used in production of beer and other different products. So these are some uses of fungi. Now let's look at modes of nutrition in fungi. Fungi follow different modes of nutrition, and they are usually heterotrophic living organism following heterotrophic nutrition. They can't synthesize their own food. They are dependent on others for food. What fungi do? Fungi usually secrete enzymes outside their hypha, hyphae. So outside the fungal body, fungi excrete some enzyme, and those enzyme will digest the dead and decaying organic matter. And after the digestion or breaking down of the organic matter by the enzyme released by fungi, there is breaking down, and those broken down pieces are absorbed by fungi. So fungi are absorbing soluble organic matter mainly from the dead substrates. So dead substrates here refers to the dead organic matter, which is food for the fungi. So fungi follow saprophytic mode of nutrition. Some fungi follow saprophytic mode of nutrition. That means they are feeding on dead and decaying matter. So when we keep our bread for a long time, due to moist condition of the bread and because of rotting, there is uh, decaying, there is growth of fungi called rhizobus. So along with that, on rotten fruits, many different fungi usually grows. So fungi are saprophytic mainly because of they are feeding on dead and decaying organic matter. Some fungi are parasitic in nature. They do depend on others for food. Usually fungi called albugo, which are parasitic on mustard. So the host of host plant of albugo is mustard plant. So from mustard plant, albugo receives nutrition, receives nutrients. So fungi follow saprophytic and parasitic mode of nutrition. Along with that, some fungi are symbiotic. Symbiotic here refers to fungi associate with other living organisms. Say in case of lichen, there is association of fungi and algae. So you have seen lichen commonly found growing on trees and on rocks. If the place is non-polluted, then there is growth of lichen on trees, on rocks. So we also have other example called mycorrhiza. Myco refers to fungi. So fungi associate with rhiza. Rhiza refers to roots. So fungi associate with roots of higher plants, higher trees, say like pine trees. In the roots of pine tree, if you observe, then there is growth of tiny hair-like structures, and those are fungi. So fungi associate with roots of higher plants, and that forms a symbiotic association of fungi and roots of higher plant, and that is usually termed as mycorrhiza. So fungi follow saprophytic mode of nutrition, parasitic mode of nutrition, as well as symbiotic mode of nutrition. Now let's discuss about different modes of reproduction in fungi. Fungi usually follow vegetative mode of reproduction, and they also show different other mode of nutrition. First, let's look at vegetative mode of reproduction in fungi. So, fragmentation is one of the vegetative mode of reproduction, followed by some fungi. In fragmentation, there is breaking down of fungal hyphae, and from the broken pieces of fungal hyphae or the fragments of fungal hyphae, there is production of new fungal body, and that is called as fragmentation. And some fungi follow fission. Fission is splitting of fungal cell into two cells, and from which there is development of more fungi. So fungal cell will split into two cells. First, karyokinesis occurs. There is division of nucleus, and then later on division of cell occurs. Cytokinesis occurs, and then there is formation of two cells. So two cells are formed by splitting of the parental body of fungi is known as fission. Some fungi follow budding. Budding is commonly observed in yeast. So yeast, on the body of yeast, there is production of tiny bud, which will be detaching out, and there is formation of new yeast. You have seen that in yeast, first there is formation of bud, and to that bud there is distribution of nucleus. And after the distribution of nucleus, after the karyokinesis, then there is cytokinesis occurs by detachment of the bud, and then there is formation of many fungal daughter cells. So some fungi follow vegetative mode of reproduction by fragmentation, fission, or by budding. Fungi also follow asexual mode of reproduction. In asexual mode of reproduction, fungi usually produce asexual spores. One of common asexual spore is conidia. Conidia are usually produced on conidiophores, a stalk on which there is production of conidia. Sporangiospores. Sporangiospores are usually produced in sporangium, and these are asexual spores. Another asexual spore is zoospores. Zoospores are usually motile. They can move from one place to another, and mainly because of the presence of flagella. And these zoospores are produced within the zoosporangium. So zoospores, sporangiospores, and conidia are asexual spores. So asexual reproduction is usually carried in fungi by spore formation, by formation of asexual spores. Some fungi are versatile. They can change the mode of reproduction. They can become they can become vegetative reproducers, and sometimes they reproduce by asexual mode of reproduction. And in during adverse condition, in order to adapt with the nature, they reproduce by sexual means of reproduction. So sexual reproduction is mainly done by production of sexual spores. Sexual spores can be oospores. Oospores are produced within the oogonium, or they may be producing ascospores. Ascospores are produced in ascus. There are some fungi which produces basidiospores. Basidiospores are produced on basidium. So fungi reproduce sexually by formation of spores like oospores, ascospores, and basidiospores. Some fungi follow sexual cycle of reproduction, which is usually a three-step sexual cycle. First, there is occurrence of plasmogamy. Plasma refers to protoplast. Gamy here refers to fusion. So fusion of two fungal protoplasts, two different hyphae coming together, and fusion or mixing or merging of the fungal protoplasts is known as plasmogamy. After plasmogamy, nucleus of these two different fungal hyphae or fungal protoplasts will fuse together, and that is called as karyogamy, fusion of nucleus. Karyo refers to nucleus, right? And after the formation of a common cell, that is called as zygote, then there is a meiosis occurring in the formed cells. Meiosis is followed by the formed cell called zygote, which will later produce haploid spores. So spores are produced from the formed zygote. So these are the three-step sexual cycle of reproduction in fungi. First, plasmogamy, fusion of protoplasts. Second, karyogamy, that is fusion of nucleus, and then there is formation of zygote, which will be undergoing meiosis and produce haploid spores. So this is a general overview of different modes of reproduction in fungi. We'll discuss separately about reproduction in separate classes of fungi. Now, we have different classes of fungi grouped under kingdom fungi, and the basis for division of these grouping of these different classes are mainly based on morphology of the fungi and morphology of the mycelium, which will be making up the fungi. So morphology of the mycelium can be of different types. Some fungi are having septate mycelium, some are aseptate, right? So on the basis of morphology of the mycelium, we have different groups of fungi. Or on the basis of different mode of spore formation, fungi are categorized. Or on the basis of fruiting body, some fungi are having sporangium as a fruiting body, some are having basidiocarp, some are having ascocarp. So on the basis of presence of different fruiting body are structures which are producing spores. So on the basis of different fruiting body, we have different classes of fungi. So classes of fungi can be of Phycomycetes, a class of fungi, Ascomycetes, Basidiomycetes, and Deuteromycetes. So we'll discuss each one of these classes one by one. First, let's discuss about Phycomycetes. Phycomycetes are commonly called algal fungi, mainly because of their resemblance with the algae. So members of Phycomycetes are found in aquatic habitats. They are found in water bodies or on the decaying wood as saprophytic. fungi. Or they are also found in moist and marshy places, damp places, places with high water content, or some of the members of Phycomycetes are obligate parasites on plants. Obligate parasite here refers to they are uh, totally dependent on the host. They can't follow other modes of nutrition. So, on the other hand, we have facultative parasite. They can they are versatile. They can be parasitic as well as they can follow other mode of nutrition. But obligate parasites are dependent on host. They can't follow other modes of nutrition. Mycelium of members of Phycomycetes are aseptate and synocytic. Aseptate here means not having cross walls in the hypha, and because of the absence of cross walls, nuclei are distributed in many nuclei are distributed in cytoplasm. That's why they are having multinucleated hyphae and called synocytic. Fungi included under Phycomycetes reproduce asexually by production of motile spores called zoospores, and some of them produce non-motile spores called aplanospores. Zoospores and aplanospores are usually produced within the sporangium. So sporangium is a bulge structure represented here attached to the sporangiophore. So a tiny stalk called sporangiophore. So these sporangium will be releasing either zoospores or aplanospores. Zoospores are motile due to presence of flagella, and aplanospores are not having flagella and they are non-motile. We have another sexual uh, type of spores in the members of Phycomycetes that is called as zygospores. Zygospores are formed by fusion of two gametes, two different gametes, male and female gametes. So over here shown that they one fungal hive, one part of fungal hypha and other part of fungal hypha fuse together forming a spore, and that spore is called as zygospores. So zygospores can be of different in morphology. Some of the zygospores or sexual spores are usually similar in morphology, having same size, same shape, structure. That's why they are called as isogamous. Iso means same, gamous means gametes. So gametes which are similar in morphology, similar in size, shape, structure, are known as isogamous gametes. Some sexual spores are dissimilar in morphology, and they are known as either anisogamous or oogamous. Oogamous are gametes which are produced in oogonium. Anisogamous are gametes which are having different physical structures. So isogamy is fusion of isogamous gametes. So fusion of isogamous gametes is known as isogamy. Anisogamy or oogamy refers to fusion of anisogametes, gamous gametes. Oogamy here refers to fusion of oogamous gametes. Oogamous gametes are produced in oogonium. So we have studied that sexual spores can be isogamous, having same morphology, or anisogamous and oogamous, having dissimilar morphology. Fusion of isogamous gametes is known as isogamy. Anisogamy is fusion of anisogamous gametes. Fusion of oogamous gametes is known as oogamy. Examples of Phycomycetes can be Mucor, Rhizopus, that is also commonly called as black mold, Albugo, which is parasitic on mustard. So this diagrammatic representation is making you quite clear about the different sexual mode of reproduction, a different mode of reproduction in rhizobus. Rhizobus usually grow on old or rotting or decaying bread. So rhizobus follow asexual mode of reproduction as well as sexual mode of reproduction. In asexual mode of reproduction, rhizopus usually produces spores inside sporangia. So within the sporangia, there is production of spores. So these spores will be germinating and they form hyphae, and later hyphae join together to form mycelium. And hyphae can be of different types of different type of hyphae formed are genetically different. So we some hyphae can join together, can fuse together. We have plus mating type and negative mating type. So two different fungal hyphae fuse together and undergo plasmogamy. So fusion of protoplasm of two fungal hyphae will later produce fusion will be producing gametes, which may be male gamete or female gamete. And later on, there is production of zygospore. Zygospore is sexual spore, which is sexual spore, which is produced by fusion of two different fungal hyphae, two genetically different fungal hyphae. So first, at the time of fusion, we have plus and negative mating type of fungal hyphae, which are known as gametangia. Gametangia here refers to structure that is producing gamete. After fusion, there is formation of zygospores, and zygospores will undergo meiosis. Zygospores after germination, they undergo meiosis to produce spores inside sporangium, and those spores, fungal spores, will germinate and form other fungal hyphae, and later on again there is continuation of the life cycle of rhizopus. So this is how sexual as well as asexual mode of reproduction are occurring in rhizopus. Now let's discuss about Ascomycetes. Ascomycetes are sac fungi, commonly called as sac fungi. Example of Ascomycetes can be morels, truffles. So truffles, morels and truffles are expensive edibles, expensive mushroom. So we have Penicillium, Aspergillus, Neurospora, Saccharomyces cerevisiae, which is yeast, Claviceps, morels, and truffles. And all these are example of Ascomycetes. Members of Ascomycetes are multicellular except unicellular yeast. Mode of nutrition followed by members of Ascomycetes can be saprophytic, feeding on dead and decaying matter, or decomposers. They are breaking down dead and decaying matter, and that's why they are decomposers. Parasitic, dependent on host living organism, or some of the Ascomycetes. Members of Ascomycetes are coprophilous. Coprophilous here refers to growing on dung of different living or different animals. So some fungi grow on dung, and some fungi of Ascomycetes are growing on dung, and they are called as coprophilous. So members of Ascomycetes follow these different modes of nutrition. If we observe the mycelium of members of Ascomycetes, then they mycelium are branched and septate. So mycelia of members of Ascomycetes are branched, having many branching structure, and septate, having cross walls in the hyphae. Asexual spores which are produced by members of Ascomycetes are either conidia, or it may be other different spores. So conidia are usually exogenous spores produced outside, as you can see here, spores which are produced outside the conidio of outside the fungal hyphae or fungal body, and these are conidia, and conidia are spores, asexual spores produced outside the fungal body, and usually the conidio uh, 4, which is the stalk, to which there is attachment of a structure, and that structure will be producing conidia, fungal asexual spore. So sexual spores which are uh, observed in Ascomycetes are ascospores. Ascospores are usually produced within a structure, and a sac-like structure that is called as asci. Asci is plural form of ascus. So inside ascus there is production of ascospores, and ascus is usually arranged in a fruiting body called as ascocarp. Ascocarps are fruiting body, and in ascocarp we are able to find asci, and inside the asci there is production of ascospores. Now let's look at a general overview about sexual and asexual mode of reproduction in the members of Ascomycetes. As you can see here, Ascomycetes life cycle can follow sexual mode of reproduction as well as asexual mode of reproduction. In asexual mode of reproduction, there is production of spores. Spores called conidia are produced, which will later germinate and form hyphae and mycelium. Hyphae or mycelium can be of different types. It may be possessing ascogonium or anthridium. So male type mycelium or female type mycelium, or male type hyphae or hypha, or female type hypha. So fusion of male type and female type hyphae will uh, undergo fusion of male type hyphae and female type hyphae coming together of these two different mating type will uh, form a structure, and that structure is usually having two nuclei. So first there is plasmogamy occurring, fusion of male type mating male type and female type hyphae. So fusion of male type and female type hyphae, and that fusion of hyphae is known as plasmogamy, fusion of protoplasm, or mixing or merging of cytoplasm of these two different hyphae. So two different hyphae will come close together and fuse together and forming a common cell, and inside that common cell there are two nuclei, and nuclei will not fuse uh, immediately. So fusion of nuclei takes time. So that's why the life cycle, sexual life cycle of Ascomycetes follow a dikaryotic phase, and dikaryotic phase usually have dikaryotic hyphae having two nuclei in the uh, two nuclei. See here, red dot and blue dot, red dot and blue dot. So two nuclei are usually present within a cell, and this kind of stage is called as dikaryotic phase. And after dikaryotic phase, there is fusion of nuclei occur. After the fusion of two nuclei, after the phase called uh, dikaryotic phase, then complete fusion of nuclei occur, and that is called as karyogamy. First, plasmogamy occurs, and then there is a stage called dikaryotic phase in which cells are having two nuclei. And after the fusion of two nuclei, there is formation of a single nuclei, and that kind of stage is called as karyogamy. After karyogamy, then meiosis occurs. In meiosis, first there is production of, after meiosis, there is production of four daughter cells. So I'll explain in brief. So after meiosis, then there is production of uh, four spores, and these spores are usually exogenous, spores produced outside the fruiting body. So this entire structure represents ascocarp, and inside the ascocarp uh, we have structures called asci, which are a singular form of asci are called as ascus. Within the ascus, there is meiosis occurring, and by meiosis there is production of mini spores. And in a way, later spores will germinate and form new fungi. So the steps are simple. In sexual mode of reproduction, plasmogamy occurs, and after plasmogamy, karyogamy occurs. Karyogamy is a long-lasting step, which takes time, as first there is no immediate fusion of nuclei occurs. After fusion of nuclei, then meiosis occurs, which will later form spores. The fruiting body of members of Ascomycetes are known as ascocarp, and within the ascocarp we have asci, or singular form of asci is ascus, and ascus will be producing exogenous spores called ascospores, which will germinate and form new fungi. So Ascomycetes follow sexual as well as asexual mode of reproduction. Now let's look at Basidiomycetes. Basidiomycetes are commonly known as club fungi. Examples can be mushroom, which are technically called agaricus, and bracket fungi, puffballs. We have Usteligo, which are also known as smut, and Puccinia, rust fungi. Smut and rust fungi are common fungus, are common name. So usually members of Basidiomycetes are either growing in soil, or they can also grow on lops or tree stems. They can be also parasitic in plants. Mycelium can be septate and branched, having cross walls and the hyphae, so septate and branched. Generally, asexual spores are not observed in Basidiomycetes. They usually produce basidiospores, sexual spores. So vegetative reproduction is usually occurring by fragmentation. Members of Basidiomycetes undergo fragmentation of the fungal hypha, and then later on fragments of hyphae will develop into new fungal body. Now this is our view of sexual reproduction as well as asexual reproduction in Basidiomycetes. Now you are able to see three main stages: plasmogamy, karyogamy, and meiosis in zygospore. So we also have asexual mode of reproduction, wherein there is production of spores which will later germinate and form fungal hyphae. So fungal hyphae can be plus mating type or minus mating type. These are usually genetically different fungal hyphae. or these two have different genetic makeup. So these two different fungal hyphae will come close together and fuse to form a common uh, hyphae, and this stage is called as plasmogamy, or we can explain it as a fusion of protoplast of two different fungal hyphae. Later on, there is a stage called dikaryotic stage. In dikaryotic stage, we are able to observe dikaryotic mycelium or dikaryotic hyphae. In the fungal hyphae of the uh, dikaryotic phase of the Basidiomycetes, we are able to observe two nuclei in each cell, reddish and greenish. So you are able to see two nuclei in each of the cells in the mycelium of Basidiomycetes. So this stage where there is presence of two nuclei in the fungal cell will persist or will continue for some duration, say for some for till this favorable condition, till the favorable season, and later on there is fusion of the nuclei occurs, and that completes the karyogamy. So usually we observe mushroom that we see, that we eat, uh, in a structure similar to that. This, right? Usually umbrella shaped. Umbrella shaped mushroom is the basidiocarp. Basidiocarp is the fruiting body. And under the mushroom umbrella of mushroom, you are able to see gills. And these gills are usually having uh, basidia. Basidia are arranged under the gills of fungal umbrella, and these fungal umbrella uh, structure will be having will possess basidia. Basidia is plural form of basidium. So basidia will be producing spores, and there is production of 4 spores on each of the basidia by process of meiosis. So first plasmogamy occurred, fusion of two different fungal hyphae, and then there is a stage called dikaryotic phase in which we have two nuclei in each cells of fungal cell, and later on these two nuclei will fuse to complete karyogamy, and after karyogamy then there is production of spores by process of meiosis on basidia or basidium. So enlarged image of this site can be uh, represented here, having tiny structure called basidium, on which we have 4 basidiospores, 4 fungal spores produced by meiosis. Little easy to understand. So to make it easier, I would say plasmogamy occurs by fusion of plus and minus mating type. Then there is a dikaryotic phase wherein we are observing two nuclei in each cells of fungal mycelium, and after completion of karyogamy, after fusion of these two nuclei, then there is production of a structure. Fusion occurs within the basidium, and basidium will be later will be undergoing meiosis, and then produces 4 different uh, structures, 4 different spores called basidiospores. And basidiospores are haploid in nature, which will germinate and form hyphae. So this is how sexual reproduction in Ascomycetes follows. This is a description. Usually, I was saying Ascomycetes, right? It's Basidiomycetes. So Basidiomycetes are not having sexual organs. As I've explained, first plasmogamy is occurring by fusion of two cells fungal cells of fusion of two fungal hyphae, which can be also called as vegetative or somatic cells of two different strains or different genotypes, two different plus and minus mating type of fungal hyphae. And later on, there is occurrence of karyogamy. Before karyogamy, a resultant structure which is formed by fusion of two different hyphae form a dikaryotic structure, which will be later forming basidium. And after complete karyogamy, there is production. Later on, there is undergoing of meiosis and then production of 4 basidiospores. So basidiospores are produced exogenously, produced outside the fruiting body or outside on the basidium. Basidias are usually arranged in fruiting bodies called basidiocarps. So these are the how you need to read all these and remember all these, and do follow that diagram. Now let's discuss about Deuteromycetes. Deuteromycetes are also known as imperfect fungi. Example can be Alternaria, Colitotrichum, Trichoderma. So these are usually infectious fungi. So imperfect fungi are also known as fungi imperfecti, mainly because of they are not showing the sexual form or perfect stage is not known. If we know about the sexual stage, then usually we categorize them either in Ascomycetes or Basidiomycetes class. So fungal Deuteromycetes will not be showing proper sexual stage, or sexual form of the Deuteromycetes are not known. That's why they are known as imperfect fungi. Deuteromycetes itself refers to imperfect fungi. So once if we come to know about the sexual stage of the Deuteromycetes, then we usually categorize them in these two classes, Ascomycetes and Basidiomycetes. So usually they are reproducing by asexual spores called gonidia, and mycelium of the Deuteromycetes are septate as well as they are branched. So mode of nutrition followed by Deuteromycetes are saprophytic, parasitic, and they are decomposers. Being decomposer, they help in mineral recycling. So they usually play important role in biogeochemical cycles, like we have a nitrogen cycle, phosphorus cycle, we have sulfur cycle. So these different biogeochemical cycle, even we have carbon cycle, oxygen cycle. So all these biogeochemical cycles are having these different biochemical cycles dependent on decomposers. Decomposers can be bacteria, and fungal decomposer can be Deuteromycetes also, and we do have other fungi which are also helping in decomposition. So this is our view of all the classes of fungi. We have Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes, and these are categorized on the basis of morphology of mycelium, mode of spore formation, and fruiting body. And these are the features. So habitat, mycelium, mode of reproduction, mode of nutrition, and examples. So you need to, though I've discussed a lot about fungi, you need to understand this chart. This is the chart which summarizes almost every basic concepts of the 3 different classes of fungi. Now with this we have done discussion about fungi, kingdom fungi. Now you need to answer these questions without taking much time, you can pause the video and you can answer these questions. I prefer you to pause the video and answer these questions. If I keep on dictating, then it takes time. If I keep on explaining the questions are simple, so you need to follow the question and answer these questions. Thank you.